Okay, sounds good. Hi, everyone. It's a pleasure to be speaking with you today. I'm Lizzie. I am the Business Development Manager for AIM Biotech. And today I'm going to be sharing some information and data on how our vascular MPS models have been used to advance drug development for retinal neovascular diseases. So before getting into the data, I'm going to begin by giving a brief background on our company, since this is our first 3RC webinar since we are a new member. And I will then give just a very brief overview of our MPS platforms and some core applications. I'll then get into the main portion of the talk, which is going to focus on two case studies that use our vascular MPS models. So the first will be looking at how our angiogenesis model was used to support the development of a novel therapeutic for retinal neovascular diseases. And then the second study will look at how our vasculogenesis model was used to create an inner blood retinal barrier on a chip, which was used to model disease progression and identify mechanisms of action leading to diabetic retinopathy. And then finally, I'll summarize, and if I have some time, I'll answer a few questions. Okay, so who are we? Who's AIM Biotech? If you're not familiar with us, we were founded out of the pioneering lab of Roger Kam at MIT. And then through the support of the Singapore MIT Alliance for Research and Technology, or the SMART Innovation Center, we began commercial operations in 2015 with our current R&D and manufacturing productions based out of Singapore, led by Chimun Kwan, and supported by the A-STAR Institute of Molecular and Cell Biology. And since then, we've expanded greatly, both in terms of personnel, like hiring myself, and our commercial offerings as we've shifted out of academia and into more of the life sciences drug development industry. And so if we look back on the last 5 years or so, we can appreciate how AIM has grown to become much more than a device company. Of course, we have focused on our cultureware offerings and have modified those and advanced them to increase throughput and address new research needs. But we've also launched some new products with cell systems where we combine our platforms with standardized biology. And then we have offered strategic services and have formed partnerships to offer drug screening services, custom model development, and integration of complementary technologies. And so through all these efforts, we've been able to support more than 250 labs across more than 22 countries worldwide with a publication track record that really does speak for itself. We have over 200 customer publications across numerous human health and disease contexts. So a few of our core applications are shown here, which includes vascular biology, and that will be the focus of today's talk. And these beautiful images were captured directly from our platform, which range from our very first device, which was a 3-experiment chip, to a scaled-up version that enables 40 experiments per plate, as well as our newest platform that enables culture of larger tissue samples up to 2 millimeters in diameter. So with all that as a background on kind of who we are and what our technology is, I'm now going to share how our vascular models have been used in ocular and retinal research. So the first study I want to go through is the development of a novel drug modality to target retinal neovascularization, which was conducted actually out of the IMCB in Singapore by a different research group. So retinal neovascularization, which is the formation of abnormal blood vessels, is a hallmark of several retinal diseases, including diabetic retinopathy and age-related macular degeneration, which can lead to vision loss and impacts over 400 million people worldwide. And so the reason that this group wanted to develop a new therapy is because the first-line treatment, which are intravitreal injections, is highly invasive and expensive, and alternative treatments that use topical delivery of anti-VEGF compounds often fail to reach the retina due to the ocular barriers present in the eye that prevent the drug transport to the retina. And so this paper walks us through various in vitro, ex vivo, and in vivo studies that they conducted to develop and evaluate the biocompatibility and bioactivity of a topically delivered hydrogel-based nanomicelles that are loaded with an FDA-approved anti-VEGF compound, aflibercept. And so using this paper as an example, I just want to highlight that all of the models and assays that they use gave us interesting information and helpful information. But if you actually want to identify druggable targets against angiogenesis, a human angiogenesis model is needed. And so I'm not going to walk through all of the assays, of course, but I do want to highlight a few that just looked at efficacy. So using a wound healing assay, the researchers showed that aflibercept led to slowed wound healing, which was expected. And interestingly, that the drug carrier, the NEPCs, also independently slowed wound healing, suggesting an intrinsic anti-angiogenesis effect of the carriers themselves. And this trend was observed in a tube formation assay, as well as an ex vivo mouse explant sprouting assay. And so skipping all the way to the punchline, the researchers used an in vivo mouse model of choroidal neovascularization, or CNV, which uses a laser to essentially induce an injury in the eye and trigger neovascularization. And then they can add their treatments to see how neovascularization is slowed. And so what they showed is that the compound they were developing, which are the NEPCs loaded with the drug, slowed neovascularization the most compared to the other controls, which confirmed the therapeutic activity. And so while all these models provided us some information on the activity and of the drug, the isolation and interrogation of mechanisms of action controlling angiogenesis was not possible in these systems. And so to characterize the ability of the drug and carriers to inhibit actual angiogenesis, AIM's platform, our angiogenesis MPS model, was used as well. So in this assay, HUVECs or endothelial cells are seeded into one of the channels, and they are able to form a confluent monolayer in that channel, and then they are able to sprout and migrate into a pre-existing 3D collagen matrix under the concentration gradient of angiogenic stimuli. And so in this study, they were able to show, this was the only actual assay that showed this in the paper, was that the anti-VEGF compound, aflibercept, had the most effect on inhibiting angiogenesis as compared to the carriers, in the sense that cells were able to still migrate and move about within the collagen in the presence of the carriers. And so what this finding suggests is that if you do really want to look at true human angiogenesis, to tease out the effects of the drug compounds versus the drug carriers, you would want to have a angiogenesis MPS model. And so in future studies, you could use this model to further investigate the mechanisms of action and then identify druggable targets. But also, what would really help here too is the development of an eye on a chip with all the ocular barriers present that could be really useful for studying the PK/PD of anti-angiogenesis compounds that are added so that we can reduce the number of animal models for these studies. Okay, and so then switching gears from angiogenesis, we're going to talk about now vasculogenesis and how our vasculogenesis MPS model was used to create a human inner blood retinal barrier, or IBRB, to understand disease progression of diabetic retinopathy. So Heloise Regel from Roche had this initial idea and contacted Roger Cam, our co-founder, to request his help as an external advisor on the development of this model, which is crucially needed because more than 9.6 million people in the US suffer from diabetic retinopathy. And the reason this model is needed is because the progression from healthy eyes to this early stage of diabetic retinopathy, which is called non-proliferative diabetic retinopathy, is very poorly understood. And most patients are coming to the clinic when they have already advanced to the more aggressive form of the disease, which is called proliferative diabetic retinopathy. And so if we could have a model of these earlier diabetic retinopathy stages, we could hopefully identify druggable targets to prevent the advancement and progression of the disease. And so using the same device, AIM Biotech device that was used in the angiogenesis studies, the same device is used here, but instead the cells are added into the central channel. And so the cell types are retinal microvascular endothelial cells, retinal periocytes, and retinal astrocytes. And those are mixed with a fibrin hydrogel, which then enables these cells to form self-assembled microvascular networks forming the IBRB. So to validate the model, immunofluorescence staining was used to look at the expression of cell-specific markers, cell-to-cell adhesion, as well as the basement membrane proteins that are present, all of which are indicating mature networks with 3D lumens, as you can see in this cross-section. The researchers also evaluated the functionality of the networks by evaluating permeability of the vessels using fluorescently tagged dextran. And in the presence of a pro-inflammatory cytokine TNF-alpha, there was an increase in permeability, which would be expected. And so to create now a model of the diabetic retinopathy, a diabetic cocktail was applied to the system over the course of 28 days. The similar analysis was used of protein expression, and so that revealed how there was vascular regression resulting from stimulation of diabetic retinopathy, as well as a loss of periocyte coverage and the formation of what they refer to as ghost vessels, which are essentially tunnels that are created within the basement membrane proteins, but that lack the endothelial cells. And so in the presence of diabetic stimulation, the presence of those ghost vessels went up. And so all of these metrics serve as evidence of an early diabetic retinopathy phenotype, which is what corresponds to the damage and degradation of blood vessels, causing them to swell and leak fluid, which is what contributes to vision loss. Beyond image analysis, other readouts were used to validate this NPDR phenotype, including differential gene expression analysis, which I'm not going to go through, but I do just want to highlight that they were able to identify gene targets that are related to different functions, such as vascular regression, pericyte loss, inflammation. And then similarly, an ELISA is able to be used with this model to determine how different secreted proteins that are related to these same functions are different between healthy and diabetic conditions. And these are ultimately what are going to help inform these mechanisms of action to intervene and prevent progression of diabetic retinopathy. So sort of just in summary of these two studies, angiogenesis and vasculogenesis MPS models enable development of disease models and identification of underlying mechanisms of action for retinal diseases. And these platforms can be used to evaluate new therapeutics and identify druggable targets. And so I just want to also quick acknowledge that it can be very challenging to identify the right cell sources and biological materials that enable these vascular systems to work and become more reproducible and scalable. And so at AIM, what we have been working on in the last couple months really is integrating these cell sources that we have validated and standardized with our platforms to streamline the entire research study. And so to start, we have announced two products. One is Angio Prime, which is an assay-ready angiogenesis kit, and then the VasQ kit, which is an all-in-one kit for generating blood vessel networks. So I will stop there, just show you kind of like how we operate and what our information is here. And I just want to thank you all for your time, and I'd be happy to answer any quick questions if we have time for that. But yeah, thank you so much.
It's a pleasure to be here today. For those of you who don't know InSphero, I'll start off by giving you a quick introduction into what we do at InSphero and spheroids, and then take a little diversion into our model that we've developed for ocular fibrosis. Unlike presenting sort of case studies, what I'm going to talk to you about today is the development that we've done for a high throughput screening model for ocular fibrosis. And I had the distinct privilege today to talk to you about this incredible work that's been done by a senior research associate at InSphero, Thomas Hofstetter, who spent about 1.5 years really refining this high throughput screening assay for multiple high content imaging endpoints. And it's finally at a stage where it's been used for both the primary and a validation screen. But I will not be sharing any of the screening data because that is proprietary, but I will walk you through the development of the assay itself. So in terms of what we do at InSphero, we really are the spheroid company. And the reason I want to sort of spend a couple minutes talking about spheroids is because when in the world of microphysiological systems, as defined by most of the regulatory bodies, MPS systems have a very broad they cover a very broad space, right? Anything from spheroids to systems that have a lot of flow associated with them. But with spheroids, I think I'd like to focus on the particular on the things that they bring to the table that are sort of sometimes not really given a lot of importance. One of them, of course, is simplicity. The ability of spheroids to balance throughput and complex physiology is extremely important, especially when you want to think about robustness and reproducibility of the system. So with spheroids, what we're able to do is really generate data that is robust and reproducible in different laboratories around the world while being equally predictive as some of the more complex models. And this is exactly what this ocular fibrosis model will speak to you about today, because it's compatible not only for primary screening, but also for confirmatory screening in medium to high throughput models to really build a blueprint of phenotypes for both disease as well as toxicity and safety pharmacology models. In terms of applications for spheroid screening models, not only are they very applicable in the early sort of drug discovery stages in terms of understanding mechanism of action and predictive and investigative tox, but they're also being increasingly utilized in clinical stages as well when you go from sort of phase 1 to phase 2B for preclinical biomarker assessment and sort of translating into clinical relevance when you're talking about being able to stratify patients for different groups when it comes to going into from phase 2 to phase 3. So when it comes to our 3D Insight platform, across all our different groups at InSphero, what do we offer as compared to all the different MPS systems out there? Now, here I will say, I think every microphysiological system has its own place, and every one of them brings its own unique position to the market. Every one of them offers something that is different. And I think where we have to get to in the microphysiological systems ecosystem really is to really understand how these systems work together and what information all of them working together can give the end user. So in terms of the 3D Insight microtissues, what they're giving to the end user really is the reproducibility and the robustness that a lot of higher order systems cannot give. They're also very useful in terms of being able to provide that automation compatibility, which you don't normally get with a lot of other systems that are much more complex, as well as the ability to get a lot of information from one well. So the ability to multiplex, which you're going to see in the high throughput screening assay that I'm going to describe today, is something that is invaluable. And in this day and age, when we talk a lot about developing algorithms using training data sets, the ability to use spheroid models to generate a lot of that predictive data on which we're going to make decisions is a big unique selling point for using spheroid models, especially with the FDA Modernization Act 2.0 and then sort of the following on the heels of it with 3.0 version. As we move into defining the context of use of each assay, this being able to understand the predictability and the information that comes out of each particular spheroid model becomes extremely important. So as I sort of walk you through our ocular fibrosis model, what I'd like to start off with is what were we trying to do and what was the unmet medical need that we were trying to address. This was done with in collaboration with a pharma partner, and their remit here really was to develop a 3D model for ocular fibrosis, and which comes with a huge medical burden, right? So age-related macular degeneration will affect about 288 million people by 2040. And there is currently no approved treatment for this. And over time, it does lead to a loss in vision. So we came up with with sort of a plan to develop a platform with this company for a screening model for ocular fibrosis. So what did this plan really involve? It involved 4 different phases. Phase 1 was the development of the actual microtissues. Phase 2 was the disease modeling and endpoint establishment. Phase 3 was refinement of the model, and phase 4 was the actual drug screening. What I'm going to be talking to you about today are phase 2 and phase 3, not going into phase 1 or phase 4. Before going into sort of the actual disease model, I'd like to spend a little bit of time talking to you about the actual biology. And this is important because this is an extremely complex model, and it's important to sort of understand the in vitro to in vivo translation. While there are many cell types in the eye that are involved in acute macular degeneration, the one that we were modeling was the retinal pigment epithelium. And so the biggest functional importance here in the RPEs really is to be able to biologically or physiologically model this apical basolateral membrane polarity. And the reason for this is because this is the one that has sort of the highest clinical relevance, be it phagocytosis or nutrient exchange, be it being able to replicate the blood-retinal barrier integrity, or the directional secretion of factors, because all of these have clinical relevance, as you can see on the right-hand side of the column over there. So as long as we were able to replicate these 3 important physiological functions of the apical basolateral membrane polarity between among the retinal pigment epithelial cells, the model would be deemed a functional representation for acute macular degeneration. So what we decided to do was then sort of an in-depth analysis of the 2D models that were already available, and then what were the gaps in those 2D models that we wanted the 3D models to fill, so to speak. And then based on those gaps, there are some 3D bioprinted models that also had some gaps. So we wanted to assess those as well, and then develop a 3D spheroid model that took the gaps in the 2D models, took the gaps in the 3D bioprinted models, and then brought them together to fill all the gaps that existed. So as you can see here, in the fibrosis that develops in acute macular degeneration, you have an EMT, right? You have an epithelial to mesenchymal translation. And if you see this 2D retina image that you see on the left-hand side, when you see this decoupling of the RPE cells and the differentiation to the myofibroblasts, what you see on the right-hand side when you actually culture them on a 2D plastic is a very linear representation of what would actually happen in the retinal pigment epithelial layer in your eye. However, if you actually culture this in a 3D cell culture, you would see a much more representative picture of what was happening in the retina, with the ability to see the degeneration of the retina, the loss, what would eventually mirror the loss of vision, the formation of the scar tissue, which you wouldn't be able to see on plastic over here, things that you would not be able to catch in the 2D model or in the 3D bioprinted retinal model, which is in fact a 2.5 sort of 2.5 printed tissue. So the difference that we sort of like were able to fill with the 3D model that we were not initially able to sort of fill with the 2D and with the 2D model and the 2.5 bioprinted model was that we were able to create in a spheroid model, a polarized tissue-like spheroid. We were able to develop this complex tunable ECM interaction that I'm going to be talking about, how we developed that. There was realistic complex signaling. We were able to emulate the physiological gradients. The clinical predictivity was high. That is data that I'm not going to be showing today because it's proprietary. The throughput was medium to low. It's going from a 96 to a 384 well format, and the cost is sort of reasonable at this point. So we started off with evaluating the model. So what did that really involve? It involved starting off with iPSC-derived human retinal pigment epithelial cells and developing the microtissues from there. And this is sort of phase one, which I won't be discussing. We used the iPSC-derived RPEs to develop the microtissues, and then used a proprietary cocktail to induce fibrosis in these microtissues. Once we developed the fibrotic microtissue, for which we had certain biomarkers, we then had a group of compounds that we know should reverse the fibrosis that was actually caused, that we were actually seeing in this ocular fibrosis model. And then we had endpoints that we were going to use to quantify not only the fibrosis, but the reversal of fibrosis as well. So what we started off doing was we started off this entire study with high content imaging. And as you can see here on the left, you see a healthy control with a Z-stack initially of about 60 microns and then a 5 to 10 micron slice. As you can see, in the cavity, you have an epithelial monolayer and core with no collagen 1 or fibronectin signal. And there's sort of like a honeycomb structure of the cytoskeleton. But when you actually induce fibrosis using the cocktail that we do, you don't see a cavity anymore with that incredible fibrotic sort of structure. You see a high collagen 1 and fibronectin signal in the microtissue and a much more disorganized cytoskeletal structure. When we add the anti-fibrotic compounds, what you can see on the top, sort of on the panel on top with the fibrotic control, is 3 out of the 5, actually three representative compounds cause a reasonable reversal of the fibrotic phenotype for the fibrotic control. And with the healthy control, of course, you would not expect to see any reversal. However, we did have one compound, and I'll show you in the next slide as well, that did not really behave as it was supposed to. Fibrophilic acid, and going down the line, we discovered why. It seemed to have a completely different mechanism of action with respect to the signaling of the compound, which is why it working through that particular signaling pathway, it was not reversing that particular fibrosis in that model. So in terms of the model itself, at this point, we did have significant disease inhibition with respect to the high content imaging endpoints that we had for collagen 1, fibronectin, and phalloidin on day 10 of the assay. So we had a working ocular fibrosis model with HCI staining and quantification for 5 out of the 6 reference compounds, but it was still very heterogeneous in terms of the readouts that we were getting. So it was a very weak induction of the disease in terms of induction of disease from baseline. So we decided to go ahead and try a couple tweaks in terms of induction of the disease phenotype to see if we could get a bigger baseline of induction, a bigger disease induction from baseline. And we used another proprietary sort of cocktail of compounds, again, using the same procedure for the production of microtissues, and then using a different procedure for disease induction for the same time period. And we used additional induction of cocktails, an additional induction with a separate cocktail of compounds, and then we use the same set of compounds for reversal. And what we did see here was that there was a strong improvement in the fibrosis with these alternative cocktails that we were using. Not only was there a beautiful improvement in the fibrosis itself, but there was also a beautiful improvement in the reversal of fibrosis from the fibrotic compounds that we were using. So what you can see here on the left-hand side is a really nice separation of the standard, the toxic, and sort of the endpoints that we ended up using for the ability to separate fibronectin that was then normalized by nuclear count, a separation that we were not getting a good Z prime factor for the assay for in the original model. And then the other aspect of something that we were not really able to quantify with a good Z prime in the original model was the ECM for collagen and fibronectin. So this, again, because we were able to increase the signal from baseline with this new cocktail, we were able to quantify collagen and fibronectin much better with this new model. And again, as you can see here on the left-hand side, we have the Z factor for collagen normalized by nuclei count, as well as for fibronectin. And we ended up getting a much more reproducible assay, as you can see by the improved Z prime factors. So in summary, what we ended up doing in terms of developing an assay was to develop, again, from phase 1, we spent a lot of time developing the biology and the microtissues of the assay. We then established a high content imaging assay that was very representative of the disease model, but it turned out that it was very heterogeneous. So we spent a lot of time really improving that assay because we had to use it in a screening mode. And then once we were able to do that, we were able to not only use it as a screening assay, but more importantly, use it to develop predictive algorithms to not only be able to differentiate between healthy and fibrotic phenotypes, but more importantly, to be able to distinguish in those in-between phenotypes, to be able to distinguish those mechanisms, to be able to further distinguish the mechanism of action of drugs for those in-between patients, which is where like 80% of the patients lie. So with that, I think I'd like to end with saying that that whole year and a half worth of work, years worth of work was really ended up giving us a very, very robust assay where we now have an extremely good therapeutic window for performing both a tox as well as an efficacy study in this assay of ocular fibrosis. And with that, I will end. And I don't know if I have time for questions, but if I have, I'm happy to take them. And this was the incredible team that actually did the work: Oslam, Agnieszka, Babel, and David.
Great. Hi, yes, I'm Bronwyn Tomlinson. I'm a scientist within the Retina platform at NewCells. For those who aren't aware, NewCells is a biotech company based in the northeast of England in the UK. We have various models that we use to study disease or test drug compounds, including lung and kidney. And here, obviously, today, I'm here to talk about the Retina platform and the in vitro models that we have to model this. In terms of current preclinical models that are used for retinal investigations, there is animal models and primary cells and human retinal explants, but there's gaps in what they can provide for us. So animal models, for instance, are not fully recapitulating the physiology of human retina. Sequences are not conserved. So if you are wanting to test a drug with particular sequences for the human disease, animal models are not the most applicable to be able to test these. Also, if you're using models such as rodents, they're nocturnal, so actually their photoreceptor ratios are different compared to in the human. So that makes it slightly less applicable for testing drugs as well. Primary cells and cell lines obviously do not mimic the interaction of multiple cell types within the retina. And although you can get access to human retinal explants, this is difficult to get the ethics for this kind of tissue. There's a restricted window of use, and it's also limited in terms of the tissue that you can access. And unfortunately, until you get that tissue in your lab to start, you don't know the quality of that tissue. There's kind of varying quality issues there, and that will depend on what you can do with it once it actually makes its way into your lab. At NewCells, we have iPSC-derived retinal models with the main retinal types in 3D organoids, as well as an isogenic 2D RPE cell model as well, which can be used, which are human models for predictive results. Here, just went them a little bit further. So like I said, we have our retinal organoids, which model the neurosensory part of the retina with all of the major cell types, only missing the RPE, but we do have an isogenic control RPE model, which means that actually we can model all aspects of the retina with between the two models within our retinal platform. Focusing on the retinal organoids specifically, we manufacture these at scale, which allows us to grow a large number of them. By day 150, as you can see here, they contain all the major retinal cell types, including the kind of photoreceptors, in a laminated fashion all the way through to your kind of bipolar, horizontal retinal ganglion and muller glial cells as well. They increase in size up to about 1.3 millimeters in diameter, and they're grown in individual wells and 96 well plates. We offer these in two different ways. So we in-house can provide service projects where we can take receipt of compounds or drugs or gene therapies into our labs, and we can perform them in our laboratories, or we can offer them as a worldwide shipment product. We ship all the way around the world. We use a specialized courier, which means that the organoids arrive to your lab ready to use basically once they're kind of plated back out again. Or we can do a mixture. Sometimes it's that we do service projects in-house, so we'll do some of the work and then we can actually ship those organoids back out back to clients so they can do further in-house assays as well. So it kind of is customizable in the way that people can use our retinal models for their research. Although I talked there about the fact that all of the cell types of the retina do develop, the maturation process actually is dynamic and follows retinogenesis that we see in vivo. So throughout time, there is different enrichments of different cell types. So this means that depending on the cell of interest or perhaps the disease that somebody is interested in, we can use the organoids at different stages of development. So for instance, a disease such as glaucoma, which affects the retinal ganglion cells, you may use sort of an earlier time point around day 30, day 60, which is enriched for these cell populations. Whereas you're wanting to look later at photoreceptors because you're looking at retinitis pigmentosa, you're kind of looking at the later ends of differentiation here. As the organoids develop, the photoreceptors mature, and they develop the key functional units of these cells, which are the outer segments, which we can see in bright field from day 180. They start to appear, and then they increase in number, density, and length, which, yes, we can see by bright field here, as well as the proteins seen by immunofluorescence here, as well as they kind of come and protrude up over the outer limiting membrane of these 3D structures. In terms of the organoids that we produce, we tightly regulate our differentiation. So we test each batch that we produce. We have a rolling production, which means that every month we have a new setup, which means that any one time we can provide any of the time points that are that anybody may be interested in. But at around day 150, we'll do a QC in that we'll look for the key features of retinal development. So we show that there is retinal neuroepithelium developing. We test the sterility. We also test gene expression for major genes that should be expressed at these time points, as well as protein expression, and we have tight thresholds that these must reach to be able to qualify each of our batches. In terms of the robustness of that, we quantify the number of photoreceptors, for example, in these day 150 retinal organoids. And over a large number of batches, this could actually be dramatically increased in terms of numbers of batches that we have for this data. But looking at control limits, so sort of the upper and lower limits of variation that you'd expect with any process, the number of photoreceptors that we are seeing at this day 150 time point is well within this range, which means that we have a tightly robust system for differentiating and producing these retinal organoids. Moving to the kind of the later time points, an increased maturation of these retinal organoids, the photoreceptor outer segments that I talked about and the brush borders that develop is consistent throughout batches as well. So when we've quantified how many of these organoids exhibit these, this is tightly, there's a tight expression there as well. So we have a robust system for producing these retinal organoids as well. We can use these organoids for various functional studies, so for predictive data for our clients. So this can be around kind of drug safety and efficacy screening, kind of testing cytotoxicity or whether compounds are non-cytotoxic, assessing the viability for drug toxicity. We can look at kind of how cell types change over time in developmental studies, so evaluating effect of drugs on congenital defects. We can also disease model, so we have our routine setup of wild type organoids, and we can also look to either induce a disease model, or we can also differentiate from patient-derived iPSCs, or kind of CRISPR edited iPSCs to kind of model diseases as well, which will involve full characterization, which could then, then combine kind of drug safety and efficacy with that as well. I'm going to focus on the next few slides on some gene therapy vector assessment, which is another kind of study type that we regularly carry out. So within our retinal organoids, for instance, we can test transduction efficiency. So here, both by bright field and then quantification of this fluorescence, we can see that reporter gene expression is time and concentration dependent, increasing with concentration as well as with time, with live cell imaging allowing a non-invasive measurement of this transduction efficiency. In collaboration with Oxford University, we were able to also not only just test one serotype, but we looked at multiple serotypes of AAVs as well as different promoters. And as early as 5 days post-transduction, we were able to see expression of the reporter genes. Sorry. We could see with kind of a more ubiquitous promoter that we have expression kind of all the way across all of the organoids. But if you use targeted promoters such as GRIC1, you can also see that there is kind of a particular expression within photoreceptor cells, which would be around the outer layers of the organoids, as you kind of can see in these kind of fluorescent images here, which was further confirmed with co-localization with recoverin protein, which is within the photoreceptor cells, which showed this clear ring of GFP expression in those outer layers where the photoreceptors are located. So in terms of retinal organoids, we were able to look at both healthy and diseased human retinal organoids, like I said, for characterization of what those diseased retinal organoids look like. And then we can do a variety of assays looking at drug safety, efficacy, and disease model development with morphological analysis, cell viability, gene expression, or looking at something like gene therapy, looking at transduction efficiency, cell tropism, and cell viability. We also can do a variety of readouts, looking at electron microscopy, flow cytometry, or gene expression through RT-qPCR. Like I said, the retinal organoids model the neurosensory part of the retina, and although that is not combined with the RPE, we can also offer that model as well from the same isogenic genetic background, so we can get the full picture from the same genetic background iPSCs. Our RPEs are pigmented and hexagonal in shape, growing on transverbals, and as I said, isogenic to the retinal organoids. They exhibit the key features that you'd expect from RPE, such as the key markers expressed, and they're a highly pure population, which we can functionally characterize through the development of tight junctions with an increase and stabilization of tear. We've got high levels of phagocytosis of labeled photoreceptor outer segments. And these, as was mentioned previously in the previous talk, we have this key feature that they are polarized with the expression of key proteins, either apically or basally expressed within these cells. Going back to gene therapy, it's something we can do with our RPE model as well. So throughout time, again, we could see that the percentage of GFP positive cells increased in both a dose and time dependent manner. And we could further confirm this through flow cytometry as well, which confirm the dose dependent transduction efficiency as well. Sorry about that. We can use these validated assays, so similar assays that we used before for the retinal organoids, but including ones that are specific to RPE, such as kind of TM and phagocytosis. So again, we can look at modeling diseases as well as kind of assessing gene therapy as well. Although we can do standard assays, we can also help develop custom assays as well for whatever clients may be interested in. So in terms of the retinal models that we offer here at NewCells, we can look at the full spectrum of cell types that are within the retina, which are providing a useful in vitro tool for efficacy and safety studies, disease model development, and gene therapy assessment, because they are physiological relevant predictive models which have human background, contain the main retinal cell types, and are functional. We have unlimited supply, we generate at scale, and can ship the organoids internationally, as well as, like I said, do some of the work in-house as well, with extended windows of use compared to some of the other models that are available preclinically. And importantly, we can look at healthy or disease-specific tissue. We have relevant controls and reliable study outputs. Kind of a whistle-stop tour through that, but thank you very much for listening. If you want any more information, visit our website. We have our email address there, and I'll be very happy to take some questions.
Thank you so much, Bronwyn. That was fantastic. Just taking a look, and I don't see any questions yet. But because that was our last presenter, we will have time for a bit of Q&A. I see someone's hand up. If you don't mind just typing your question in the Q&A or chat, just because we keep everyone on mute, that would be great. I see a few hands up. So yeah, just use the Q&A in the chat to type out your question, and we'll give a few moments for that. But if all our presenters want to come on screen, that would be great. seeing if I think I can actually unmute if people don't want to type out their question in the Q&A chat. So let me see. There's someone with their hand up. Okay, just give me 1 second and I will unmute. Okay, Shivarju, you should be able to unmute and ask your question.
Yes, can you hear me?
Yes.
Hi everyone, thanks for the great presentation and also the great work. I have maybe a silly question. So these organoids, ocular retinal organoids, especially for looking for a toxicity studies, this organoids retinal wall is too heterogeneous. You mean one side is thick, one side is little thinner. It's not uniform. The first question. The second question is how do we translate what we do in a toxicity study with these organoids? The second question is fibrosis, as well as we are trying to recapitulate the retinal situation. So in these organoids, you will have RPE cells within the organoid, not the monolayer. It's basically they are integrated into the epithelial system. How are you guys going to separate that patch of a new cell types within the retina, the effect of RPE cells within the retina? How are you going to separate it?
Yeah, they're great questions. Thank you. To answer your first question, so we, the organoids as they develop through do get slightly more heterogeneous. We look for the regions that are the developed neuroretinal regions, which are thick, which are the thick laminated regions. They're the kind of the areas that we would focus on in terms of analysis. So that's where we would kind of focus analysis on there. We do sometimes see the development of RPE within the organoids. You can see them, they are pigmented. It's not uniform. So it depends on the study whether we'd include organoids that have RPE within them already, because as you say, it's sometimes hard to tease out exactly what's going on if they are present. So we can kind of cherry pick which organoids look more. So we can either include them with the RPE or not. We can see them; they come up as
group.
Yeah, they kind of grow off on the side. They don't come up, and they're not in the right physiological place either. So that's why we kind of have our separate model, which kind of models exactly the RPE and kind of the physiological state more that they would be in rather than in the organoid. Okay.
Can I add a little more if I have some time? Can I add one more question?
Yeah, no problem.
If we consider physiologically relevant model, let's say retina with RPE cells, a monolayer in the bottom, it totally behaves differently, that system, than just using the retinoids. So RPE cells, when they are growing versus they are polarized, they are like a different avatars. They do different things. Now, how do we translate whatever we do it in retinoids, like organoids, to in vivo situation? Because we don't. And the second question is, do you guys have anybody is establishing these retinoids on top of a RPE cells monolayer?
It's the dream, right? That's what everybody wants. It's something that we've looked into. It's very technically challenging. There is one paper out there that's done it, and there's been struggles by people to recapitulate that. So it's no small task to do this. It's something we look at in-house. If anyone would like to help develop it with us, we're very happy to look at collaborations. But yeah, it's that is the most physiological relevant situation that we would have. It's not something we have now, unfortunately. We have to use the model that is most appropriate for the question. So if it is more RPE based, then we look at the RPE model, and if it's looking at specifically the photoreceptors or the retinal ganglion cells, that's where the organoids are more appropriate. So it's using the best tool that we have for the specific question that we need.
Madhu, if you wanted to have anything to add, or Lizzie as well, feel free to pop in.
Yeah, and I completely sort of want to support Bronwyn's position here. I think we have to use the tools, you know, to answer the questions that we are asked. But if you're interested, Shivaraj, I think if you look at Dr. Kapil Bharati's work at the NEI, he's doing exactly sort of what you're asking about, right? And I think academics have that flexibility of looking at those things. He's bringing some of the first transplanted therapies for dry eye syndrome into the clinic. So you might want to look into what he's doing, because he's doing a lot of what you're asking about.
Thank you.
I just wanted to add too, I'm not an expert on eye anatomy, but if there's a need for having these retinal organoids adjacent to, next to, or abreast of a RPE monolayer, you can use microfluidic platforms, whether it's AIMS or not, but you can use those more compartmentalized platforms to structurally kind of create the model you're going for. So you can inject those types of cells and try to get that topography down. It's not something that AIM has done, but it's, I think it would be feasible.
Thank you.
Awesome. Thank you all for that. I don't see any other questions coming in, but I kind of have a general question that maybe each of you can speak to for your company, kind of related to like what's next for your Retina and Ocular MPS that you're working on right now. Are you kind of focusing on refining your current models? Are you, do you have any other projects that are up and coming kind of related to this? Obviously, what you can speak to, but I'm just curious kind of for each of you.
Can I come in for a second? I think it's a relevant question, what you said right now. Is any of your companies are looking for inner limiting membrane generating on top of the retina?
Yeah, so maybe if someone wants to start with, if you are looking into what Shivaraju was curious about, and if not, maybe if you want to expand upon something else that is kind of up and coming for your company. So maybe, I don't know, Madhu, if you want to start.
No, happy to start. So no, we're not looking into that, but in terms of expanding our portfolio, I can definitely speak to that. So as far as the ocular fibrosis for AMD model is concerned, we established it as a screening model, right, to get that therapeutic window, because it was to fill a particular question for a particular partner. But what we're using it for now, refining it for now, is really to enhance our fibrosis platform, which will be model agnostic. So we already have a MASH platform. We have a cardiac fibrosis model as well. So we're sort of moving really into the fibrosis area. So I think in that context, we'll be refining more of the fibrotic endpoints to build out our fibrosis portfolio.
Great. Thanks, Madhu. Lizzie, if you want to go next.
Yeah, sure thing. So AIM is not actively working on retina-related projects at the moment, but a lot of the work that we do develop in-house and that we have available lends itself to these applications, as I mentioned, through the publications I was going through. So, we're happy to support development needs, whether it's building a whole new model or just helping support onboarding and tech transfer of our models into your labs. Specifically, if you're interested in some of those vascular processes related to neovascularization. So, happy to support through any of those means by providing the cultureware or by actually doing service projects with anyone who would be interested.
Great, thanks. And then Bronwyn.
Yes, in terms of like in a layer and in a limiting membrane models, it's not something we're specifically looking into, but we can work with clients who are interested. We can help co-develop models. I've talked a lot about kind of efficacy and toxicity and testing of of drugs or like gene therapies. But we do a lot of bespoke work with clients about developing new models that they are interested in. It depends on the market demand, obviously, if we were going to do something more in-house, but we're happy to work with anybody to kind of develop the models that they are looking at, which would, you know, serve whatever purpose they need.
Great, thanks. Super exciting to see kind of what's coming next. Don't see any other questions incoming, so maybe I'll ask another one. So as we talk about often that benchmarking and standardization are important next steps for, advancing widespread use of microphysiological systems. So I'm wondering if each of you can, tell us a little bit about any published benchmarks or standards that are specific to retina or ocular MPS, either like published or maybe unpublished. If there's anything that either of you can speak to. Feel free to jump in.
I guess I could just say, we're not necessarily working on benchmarking for retina specifically, but we are, of course, looking to standardize the cell sources and the platform that we have. So in terms of the MPS platform, it's engineered in such a way where you don't need pumps, you don't need plate rockers. Gas exchange occurs actually through the bottom of the plate, so it can just sort of sit in your incubator. So its format is amenable to automated liquid handling and imaging and all of those things. So in that sense, it's a standardized plate with a 384 well plate footprint. And then, as I mentioned in the talk briefly, just looking to figure out cell sources that are consistent so that you don't have lot variability, donor variability that can come from primary cell sources. And so figuring out what are the types of cells that people are interested in, that's something that we like to ask the end users, what do you need? And we can help use our existing platforms or create a new one in order to do that. But I think it's an important conversation to have around the biology as well as the platform itself, making sure both are able to be reproducible and scalable for these questions.
Yeah, that's great. Thanks.
I think that's a great point. It's a great question, Lauren. Since our focus really was on developing sort of a medium throughput screening assay, our benchmarking has been really about getting that reproducible data, right? So, which is why once we developed sort of that phase 1 of the screening assay and realized that it wasn't as reproducible as we wanted it to be, there was still heterogeneity in the results. We went on to refine it. So I would say that sort of our benchmarking has been around the endpoints. It has been around the fibrotic phenotype for us. And I think what we focused on really, at least from the phenotype that we want to recreate for the RPEs, which is the disease phenotype for AMD, we've done a lot of validation in higher order models that, with collaborators for things that are coming out of this assay. And they seem to be very predictive of the same disease pathology. So for us, I think that has been very discerning to see.
Yeah, great. Thanks, Madhu. Bronwyn, go ahead.
Yeah, it's something we've worked on a lot in-house. It's clear that the benefits of benchmarking and creating the robustness of a model. It's something that why we have such stringent kind of QC that we do on both the RPE and the retinal organoids. We want to know that if we do an experiment on a batch now, we can do the same experiment on a batch later, and you're going to get the same results. It's important then, to be able to tease out some of these really not minute, but any of these variations you want to know is because your model is different or your compounds are causing an issue, or causing differences. It's not that there's any variation in the model that you're using. So having a fully robust system is very important to us across any of our platforms. So it's something that we work on tirelessly in-house to keep improving as well, to make sure that we are as stringent as possible, to make sure that we're getting that robust and reproducibility that we need.
Yeah, absolutely. I think great points all around. Okay, I'm not seeing any other questions coming in. So I just wanted to remind everyone before we end things today that the webinar is being recorded. So it will be sent out to all registrants as well as posted on our YouTube channel. I wanted to give a huge thank you to Maddie, Bronwyn, and Lizzie for your presentations today and for participating in our webinar. If you have any follow-up questions, please feel free to email them to me and I can pass them along to our presenters. But that will conclude things for today. I look forward to seeing you all at our next webinar in May. And thanks, everyone.
Thanks so much, everyone.
Yeah, thank you so much.